Okay, so we'll go ahead and get started. First off, how's everybody feeling?
Right.
Good? Yeah, so you just wanna, we're okay, we're not high stress anxiety. We got time to practice and work on the questions. Okay, so where do we wanna start? We have 4 potential starting points, 1, 2, 3, or 4. Who wants to go for it? We're gonna start here. You want to start here? Okay, we'll start here.
What is just a situation about talking about doing the news and why we use RNA primer instead of the DNA primer?
Yes.
I'm still not fully clear as to why we use the RNA primer instead of the DNA.
Okay, so we're going to talk a lot after exam 1 about the RNA primer strand, going back and basically getting rid of it and then filling it in with DNA, which is important for like the leading lagging strand type of thing. So there's a little bit of it in your reading, but this concept is not going to be on your exam.
Oh,
It's going to be on next exam.
that's it.
Okay, so everyone clear that this concept is not on your exam. Okay, I'm not going to ask you primase, the RNA primer, or why RNA primer as opposed to DNA primer. You can hold me to it. It's on recording. So
Next time then.
okay, but we are going to come back to it.
I do have another question, and if that was not relevant.
Yes.
So in discussion, we talked about how, there was a question that asked, what was the purpose of radio-labeled sulfur and phosphorus in the future experiment?
Mhm.
And there were there were 5 answers, but 2 plausible ones. And the first one that was selected as the best answer is that phosphorus labeled the virus genome.
Mhm.
But there was another answer in there, sulfur labeled the supermutant. And I was just wondering why we wouldn't consider that to be, important for the purpose, because we also want to know.
Right,
the
right.
protein, right?
So sulfur was detected in the supernatant, but it didn't label the supernatant.
So that would just be updated.
Yes.
Okay.
So remember, the sulfur labels the protein. So one of those things to think about as we're going through exam questions, and we're going to talk a little bit about exam strategy on Monday, because the way I ask and answer questions, it might be a little bit different than you're used to. But make sure, all right, choose the best answer and ask yourself, what is the best answer and why is this the best answer? Okay, we'll keep going down the table. Like I said, if you don't have a question, then we're happy to skip.
So my question is, for this pre-lecture quiz, for the last question, question number 8, I understand that removing the original replication causes the gene that has this antibiotic resistance not to replicate. But my question is, considering that we learned for lecture 2 that eukaryotes might have multiple origins of replication, but for plasmids they only got one. Wouldn't that mean removing the original replication would completely stop replication, even you don't have the genome out?
And so the transformation will occur, that one cell that got the gene will still live, but you're not going to have any replication. There's no origin of replication.
But the answer for this question, it takes the ones that said that it would grow on the pseudonym regardless of removing the origin of the
So I think in the question, it has the plasmid for replicating the plasmid, right? But remember, the bacteria has its own host genome. And if it's on the right media, it can still grow. It doesn't need the plasmid to grow. It needs the plasmid for expression of those selection markers, depending on the selection media. Does that make sense?
But as long as you have the original replication in the bacteria, it's all.
The bacteria itself can grow if it has what it needs. Yes. Markers on markers on markers. What colors we got?
Purple.
Oh yeah, then well, now you're into the purple markers.
Because like half our markers are dead.
Okay, 3-year-old destroyed all the purple markers.
Sorry.
It's fine.
I brought pink though.
I mean, yeah, we had pink last time, which is good.
Anything else, or you good?
That's good. Okay, right. I'll see you tomorrow.
See you tomorrow.
Okay, is that clear?
No, yeah.
Okay, so remember, the plasmid is not the only DNA or whatever material inside the bacteria or the yeast. They have their own whole genome. And if there's a situation where they don't get the plasmid, the selection markers, but you have a scenario where they have complete media or they have leucine and everything, they can still grow. They don't need that plasmid to grow. The origin replications for that particular genes on the plasmid in the context of the selection media. Okay. Question or pass? Okay, pass.
I have, question about the experiment that. Of the evidence of really happened. I understand how to draw the relative for the 200 basis points, but I don't understand how to draw it for like the 400 or 600 basis points.
Okay, so what I'm gonna do is draw two gels. Let's say this is a theoretical situation where the units are 200 base pairs. Let's do a unit of 300 base pair. Let's do units of 400 base pair. On the gel, you're already aware of this, that there was a signal at the bottom that was about at 200. There's another signal that was at 400, another signal at 600, 8. I can't math. I guess not. And 1200. And then so a lot of people, what they did was went ahead and drew their chromosomes. So chromosome, and then linker DNA, chromosome, linker DNA, chromosome, linker DNA. And drawing it next to it is actually really nice way to illustrate that every single section is another 200. It's another repeating unit of 200. So basically you would see the same thing if, for example, these units were 300. So the very first one ends up being 300, then at 600, 900, oh my gosh, 1200. Okay, look at me adding stuff. So same thing. What we don't know, the hypotheticals, is basically like how much could typically be wrapped around. We know from the size of histones that usually it's 150 base pairs of DNA wrapped around the histone core. That's like empirically basically figured out. And then so what they basically from this experiment using small amounts of nuclease, but what happened in that experiment before they ran the gel is that the small amounts of nuclease are going to cut the parts of the DNA that are not protected by the protein. So it's cutting where it's most susceptible. And the reason why you end up getting 200, 400, 600 is that sometimes it'll cut right here and you only get 1 unit. Sometimes it might cut right here and then you'll have 2 units stuck together, and that's what the 400 band is the 2 units. So does that make a little more sense if you draw it this way? That 2 units together represent that 400. Then 3 full units represent the 600. Okay.
So it's not always like every single unit is
Kind of random and where it's most susceptible. So that's how you end up getting the whole ladder. So that's a really good question. What if I wrote a problem where I said, instead of using small amounts of nuclease, I use a whole bunch, like a crap ton. What would this gel look like if I use a whole bunch of nuclease? Where do you think it's going to cut? It'll all be at 200 because it'll be so efficient that you get cut here, cut here, cut here, cut here, cut here. So you only have your little individual units because there's so much nuclease. But the stuff around the histone is still protected. Yes.
But, so the cuts happen before we do the.
Exactly.
Okay.
So that order matters. In that experiment, we nuclease first, and then we do the proteinase to get rid of the histone so we can run the gel. So this is what the complex look like when we're adding a nuclease. Got rid of the protein and ran only the nucleic acid on the gel. Okay, any other questions on this concept? And how the gel represents these repeating units? Like if I gave you a gel that looked like this, 3, 6, 900, 1200, and I asked you, what does this mean about the size of a histone? Or the histone repeating units? They're 300 base pairs. So you can figure that out, yes? Okay, good. You'll see it on the exam. Okay. Okay, next question. This one sucks. Okay, I gotta get my own eraser.
We're doing the inner row first and the outer row.
Yeah, inner row and then outer row. Yes.
I had a question. Could you just explain, the results for the third generation gradient on the small experiment?
Yes, when we're gonna do that. Yay. Okay. So this is what I mean by use 2 different markers, 2 different colors. And then, so what we're gonna go ahead and do is write first generation, second generation, and third generation. Over here, we're gonna put native and alkaline. And then we're gonna do our parental strands. See how good this brings. Our brain's pretty good. Okay, so we'll start off with heavy. parent DNA is double stranded. If we take this, shift it to our light. Nitrogen. After the first replication, we are going to have a parent strand that basically split in two and then copied itself of a daughter strand. So that's clear. They're together, so that's the native one. And so now if I separate these, the alkaline, I basically denature this. So I have two separate DNA strands here, two separate DNA strands here. So I could easily say that this is 50% heavy, 50% light. So now let's go with another generation, meaning that the blue is going to split in half, the green is going to split in half. Because we're growing it in light, we're adding a light strand to each one. So now if I go down here and separate them on an alkaline gel, I still have two of my heavy. Now I have six. I have six of my, what is this called? Light. So now we can say this is, oh my God, math. 2, 4, 6, 8, these are quarters. This is 25%, 75%. Yeah. 50%. Okay, so now I want to go to a whole third generation. I'm gonna do the same thing. I have my parent strand, oh, it's not my blue. I have my parent strand. A parent strand. Two more light parent strands. Then another 4 of these. 1, 2, 3, 4, and they all get new light. And then if you denature, you kind of get the gist. So you can see how if you take your time and draw it out, you cannot go wrong because you can visually see it. So if I ask you a question like this, at the second generation, what percent is going to be heavy, what percent is going to be light? Can you tell me that? You need to be able to tell me that. At the third generation, something you notice about semi-conservative replication is that you always have some mixed band and you add in the second part. So you could only end up having the 2 different types of band, mixed and for this condition, light. And then if you look at the alkaline where you break it apart, you only have heavy and light, and that never changes. You're just getting more and more and more of the light. So patterns that you can see. Yes, question.
So would the alkaline gel also be like, it also proves that it's not dispersive?
Yes, it proves that it's not dispersive, and it also proves that it's not conservative. So you need the alkaline gel to tell the difference. Yes.
Not assuming, we always assume that the answer would be that you asked, so the function in the right.
Never assume. So I will write, we first grew this in 15N and moved it to 14N. Or we reversed it, we grew it in 14N first and then moved it into 15N. Know that whatever it is grown in first or replicated in first is going to be the parent strand. So the parent strand can be either light or heavy. This is just used so that we know which strand is which. So pay attention to that, yes.
So because I think the textbook said something like DNA normally has like 14N or something like that.
That's the normal one.
Okay, so the 15N is always gonna make 14N, like.
No, so you actively have to use this isotope in the material that's going to be used for extension of the DNA.
Okay.
So you're making experimental conditions where you introduce the isotope or introduce like the normal version of the ammonium.
Okay, I think that's why I'm kind of confused, because I guess when I think of like DNA replication, I just think about like the one strand and then it splits off and then it creates a new one, so like. How, I guess, would like a 15N strand interact with like a 14?
So it's only going to interact because we first basically grew an organism or did replication in a place where they replicated until 15N was incorporated into all the DNA there.
Okay.
Then we're going to take it out of this condition, so there's no more access to this heavy isotope. And when it replicates, it will only have access to like nucleotides that have the 14N.
Okay. Okay, that makes sense.
So you're controlling it in vitro. This is in vitro.
Okay.
Yes.
So just like I just want to make sure I understand that in terms of why we use both the native and natural ingredients. We're separating, or distinguishing between, in the first generation replication. Native is for distinguishing between conservative and semi-conservative / dispersive.
Yes,
And then alkaline is for distinguishing between conservative / semi-conservative and dispersive.
yes, it is. So one thing I want to talk about, the way you're framing it is a memorization framework. You're trying to memorize like, okay, if I use alkaline, I'm trying to differentiate between these certain ones. Want to make sure it's right, yeah, it's correct, but I'm just like saying this to your group. When you use that framework, become reliant on what you remember as opposed to like just thinking through the actual problem.
Mhm.
So when you have like a scenario that isn't the norm, then your memory is gonna like confuse you. So as long as you understand the actual process behind it, like you'll be able to be, like, even if you can't like repeat back what you just said, you understand the process, you'll be able to tell like, oh yeah, this is this person, or this is somebody conservative, like because of the way it looks.
Yeah.
Okay, so everyone's good on that. Okay, any more things on Messelson stall?
So just to clarify, I know you kind of just talked about this earlier, so the N14 that's being used in like replication, is it coming from like other DNA strands, from like a solution?
Some solution,
Okay,
so it's a solution that we give it, so
so,
that it has access to, yeah.
sorry.
Oh, no, that's good.
So if you had like a solution with N15 and you put in half N15 and half N14, would it make N15 strands?
It would make like mixed strands, and then what would happen is that you would get results that technically you wouldn't be able to interpret, because you wouldn't know like where the 15 or 14 came from.
Oh, that was, thank you.
Yes.
Does that like look like dispersive model?
It would actually look like this person model,
Okay,
100%.
Do I need to know that, like, do I need to do this process again with the conservative dispersive model? Like, are you gonna be like?
Yeah.
About that?
I will 100% ask about it. So you'll see results of this experiment, and so based off these results, is this person or conservative or semi-conservative? Yeah. Is going to be based on the question stem. So if I say whatever you grew it in first, we first started out growing it in heavy ammonium or the 15N, that's going to be the parent. So whatever it is grown in first is the parent strand. Any other questions about the install? You need to know it, you need to know it. Good to use. Okay, on to the next. On to the next, on to the next.
So I had a question about, just like growth colonies, can you just like explain, like what makes a large colony and what would make like just more colonies in general?
Yes. We'll start off talking about transformation. But it's kind of assumed knowledge that I want to make sure is clear is that transformation is never 100% efficient. So say we have a LEU delta strain, you want to transform with a plasmid that is replicative competent. And so on the exam, I will be very clear to say, assume all plasmids have everything they need, like a centromere and origin replication, unless it's specified in the question that it's missing something. Okay, that's clear. It'll also be in the instructions on the exam. Read the instructions, please. Okay, so we have a complete plasmid that can do whatever it needs to do. And this carries like the LEU to G. And then what happens is that you're going to have like all these bacteria. You're going to have different bacteria that we try to transform with this plasmid. But not all of them are going to get the plasmid. So say, for example, this got a plasmid, you got a plasmid, you got a plasmid, everybody gets a plasmid. So these amounts get a plasmid. And then what we want to do, of course, is use a selective media, meaning that we're going to grow it without leucine. The ones that don't get a plasmid are going to die off because they don't have leucine, they can't live. So you've selected. And then the reason why we get a certain amount of colonies is because you don't have 100% transformation efficiency. The transformation efficiency, for the most part, is going to be one factor that dictates how many colonies are on the plate. The other thing that dictates how many colonies are on the plate is just how much bacteria or yeast, whatever you put on there to start with. So that's not going to be an exam, but those are the two factors. How much I put on there, how many of them actually get transformed. So now that you have them transformed, you think about what makes me have a small colony versus a large colony. So think about these as small colonies. Let's say these are some nice large colonies. And these large colonies have like plasmid. The difference being that small colonies are generated by plasmids that don't have a centromere. So think about one cell. I got the plasmid, and then I'm trying to divide and make more of myself. If I'm dividing asymmetrically, one of me didn't get the plasmid. So one of me in the selected media is gonna die off. But on the other hand, over here, every time I divide, both are gonna live. So each colony is getting bigger more and more rapidly. And here the colonies are getting bigger, but it's taking forever, so the colonies are small. So that's why having a centromere in the plasmid makes a difference in the size of the actual colony. Okay, is that clear? Yes,
So, with like the leucine, only no centromere, no origin replication on the plasmid, you get no colony, right?
okay, say it again.
So like with the leucine only for transformation,
Leucine only,
no centromere, no ARS.
But
You
there's
just get no colony. right?
no colonies.
Right,
So
then-
there's no start site, no colonies.
Plus the ARS, you get some that would survive, right? Because
Okay, so if we,
that leucine, that is being transported to some, but it's not being distributed to the mission.
Yeah.
So you get smaller.
so you said plus leucine and plus ARS. Who else is gonna grow if there's leucine present? The original delta.
Yeah.
So all these is also gonna grow. And they're gonna grow a whole bunch. So this is actually gonna result in a lawn. When you don't have a selected media,
No, the leucine gene transformed.
they transform in.
Yeah, no, not growing on.
Okay, so no leucine. That's your question. That's a question.
I'm just taking the three factors.
Yeah.
And breaking them down.
Okay.
We can separate them.
Media has no leucine. So only these can grow once they're transformed. You say with or without ARS, what's your hypothetical? With ARS, you have a start site,
Yeah.
but no
You
centromere,
know, centromere.
no centromere, that's your small colonies.
Yeah. So then, but when you combine them all, then you get six, you get selected.
Selection and large. Okay, good. Any other questions?
Well, I think it's like, the having of a centromere allows, like, a development to grow faster, like.
So having centromeres allows the genetic material to basically separate into two daughter cells equally every single time inaccurately.
And then what they dictate the colony size is, there's the efficiency rate of transformation.
Yep, and how much bacteria or yeast or cells we put on there in the first place. That's the number of colonies. Okay,
I'm sorry.
no, hey, pause, look, don't apologize for asking questions. This is my job, and the whole point is to learn. If I do everything, we're gonna be having office hours. Okay, well, let's ask questions, don't be apologetic, just get your questions answered.
Okay, so for definition of transformation, then it's not just the DNA is taken up into the new bacteria, like the one that doesn't have it, but it's also that it's successfully replicated, right?
No, so there's two separate things.
Okay.
Transformation is one step, it got taken up into the cell, replication is the whole nother step.
Okay.
Keep them separate.
So even DNA though, then that has been cut up can still be transformed into the new bacteria, but as
Mhm.
long. So, okay, so transformation then in our class just refers to when it gets taken up.
Yes,
Through the walls.
exactly,
Doesn't mean that it's successfully replicated.
exactly.
So plasmid without an origin of replication is still, like, it's still involved in transformation.
Yeah, so,
Can't
so
replicate.
transformation can still happen.
Right.
But then experimentally, like, oh, we can't check it because it didn't grow. Okay, good, notice differences,
I think, so if I have no origin of replication, I get them colonies.
correct.
And then if I have no ORI, but I have a centromere for a stable colony, so these are like.
Nope.
Okay.
You need the origin of replication.
Got it. And then I thought you said earlier that the centromere, dictates the size of the colony, not the boundary of the colony.
Exactly, so centromere is the size.
Okay, I just wanted to be sure.
Okay,
the
that's
last-
correct, 100% correct. Yes,
I know that you said that without a centromere, you produce like smaller colonies. I'm kind of confused on that because if it's not splitting up and replicating, like you don't get even amounts. That would mean that all of the material is going into one cell?
If somebody's getting double the amount of plasmid,
So wouldn't that mean there's like a bigger colony?
it's the same cell, it just has double the amount of plasmid,
Right,
but you're not getting the same number of cells.
okay,
So when you're splitting up and half of every time you divide, somebody's not getting something. Or 30% of the time, either way, it's lower, you're not replicating as fast. This is gonna amplify very quickly because every single cell is gonna double, so you get big
Okay.
colonies, and this isn't doubling at the same rate. Because we're losing any cell that doesn't get the plasmid.
okay, I see.
Okay, we'll go here then here.
Wait, so I go back to your question, thinking like, this is the original one,
Mhm.
and it's splits into two, but this one doesn't have it, so it's just one again.
It's just
Right.
what, yeah, well, you have the original parent, and then, yeah, then it's just one.
Okay.
Okay, that hand back there. Yep, so increase fish to transformation, or start off with more cells on the plate. Okay, where are we at in the circle? The circle of life. Okay,
One last question to add on, like, when you said something dictates size of like, on each individual sellers.
how big each colony is.
I think would be. I don't know, really. It's the following will increase the TM of the given base pair, and then I understand how increasing the percentage of increasing base pair makes it stronger. But I didn't understand, could you explain that increasing the doctor concentration from the 5 millimoles NaCl to 15 millimoles NaCl?
Yes, so I'm gonna explain it, but I'm also gonna reiterate, I will not ask you about salts on the exam, because it is very context specific. Everyone's clear on that. Salts will not be on the exam for how it impacts melting temperature. I want you to know that it's a factor that can. Yes, good on that? Okay, so the salts basically have the ability to stable, because you have these positive charges from the salts, they can interact with the negative backbone of the phosphate group, almost act like a protector, so that you're pushing away water from the molecule and driving those two molecules together, so it's more stable. So that happens with salts. But like I said, there's many scenarios where this actually doesn't occur, and salts can make it worse. But generally speaking, you increase salt concentration, you increase the melting temperature of the DNA. Okay. Yes,
So that was the mildly acidic and mildly basic. I like looked all over for any evidence of like it being like more stable in a mild acidic environment. I cannot find any of them.
So
So
there-
like I just found like if anything that's like not physiological pH will like aid in the nutrient, like will lower the pH.
well, it should be in the textbook. I'll go look for the pages for you, but mildly acidic actually will stabilize it. You actually find, especially in laboratories, if you're gonna store DNA, will acidify it a little bit just to store it.
Okay.
And so it does make it more stable. But of course, if you go too far down, you cause like hydrolysis and break on the backbone of the DNA, and
Yeah.
it's just destroyed.
But then the mildly basic does not do any stabilize it.
No, it'll actually destabilize.
Right.
Yes,
Why? Like the mechanism that was existing.
yes. So within acid, what is acid? What defines acid?
H plus.
You have a whole bunch of protons. What do you think a proton may react with in DNA? Multiple things, yes. So you have the phosphate backbone, and then you also have hydrogen bonds. So within those hydrogen bonds, you're basically needing to interact with the OH and a minus. So if you think about bases will disrupt the hydrogen bond, and then the acid will help stabilize that. Because you're not taking away the interaction. So bases have very strong interaction, right? So it'll disrupt the OH of the bases on the base pairs and cause the melting temperature to go down and destabilize it. But the protons instead will actually stabilize the interaction. Does that make sense? Okay, so just think about hydrogen bonding and the fact that bases are involved in hydrogen bonding. And then so replacing the bases with base on the outside is not good. Okay, anything else on that topic? Okay, next, we're still going around the table this way.
So for the mechanism of the fast that I asked you last time, I was wondering to the G and C, because I got over to the
Yes, so when we say D and P, we are just saying nucleotide. So this could be any nucleotide. C, T, A, G, U, that's what the N stands for, is nucleotide. So you'll see some specific examples, you might see general examples, this is an umbrella term. D and M P, yes. So M, mono, D, di, T, tri. Okay, everyone's clear on this nomenclature. Okay, any other question? Please talk. Yes,
I'm gonna go over the possibility, but just one of them, the mechanism of why we agree about these.
we're just gonna do this. This is P alpha. This versus beta, I can forget. Okay, and then so in order to kick off. Basically the neutrophilic attack. The 3 phosphates is more energetically favorable. For minus 2 plus to attack and kick off the 2. But what's really, really important about starting off with 3 is more than just the fact that this is energetically favorable. The idea is that if you kick off 2 of these inorganic phosphates, this reaction where you add a nucleotide is actually reversible. Let's just say this is Johnny in the worst possible way. Let's just say this is reversible. And then so we have this step where we take a pyrophosphatase. I don't want to draw it this way. It's not a good way to draw it. Pyrophosphatase. And we break this into 2 pieces. And by breaking it into 2 pieces, we are reducing the concentration of the pyrophosphates. The pyrophosphate is what could cause the reaction, which is basically adding on a nucleotide to go in reverse, which is taking it off. If we only had a 1-step reaction, there would be no way to force the reaction to go to completion. Instead, you'd have this weird reversible thing that never goes forward and you can't extend DNA. So the really important concept here is to know that you need the pyrophosphatase to break down this pyrophosphate into 2 inorganic phosphates. So now you have a chemical reaction that will go to completion and not go in the reverse direction. Yes.
Why can't the inorganic phosphate just finance it? Because you said, so, the mechanism is, you have to make it's interaction with the being 2.
Yes.
2 plus phosphate take to the top.
Yeah.
Pyrophosphate is simply into this 2, phase 2, and then phosphate.
Mhm.
How can't one of those 2 and then phosphates come back and then
Well, you would basically, so energetically, it's just not, it's not gonna happen.
Okay.
Because of the way the electrons are around, like you go on inorganic phosphate by itself. And then for this reaction, we need an active enzyme to make this 2 go back together.
Got it.
And the cells don't have this reverse enzyme. Okay, any other question? Know this concept? No, it. Okay, people around the table. You can erase this stuff with my hands.
Sorry, just about that. I was still confused, because are you still producing like the 2 inorganic phosphates, or are we just seeing that like the pyrophosphate usually somehow fine enough such that it's not going to, I guess, cause sort of this reaction as readily as if it was like produced after the first step in the reaction?
Yeah, so it produces it very rapidly and like almost like instantaneously. This is a very like strong forward reaction. And so you're always going to produce these 2 inorganic phosphates when you're elongating DNA. And then so you're not going to get the reverse at all because of the activity of this enzyme.
So those 2 inorganic monophosphates can't do the reverse reaction?
No, they can't do the reverse reaction on their own. No,
Oh, it has to be.
it has to be the diphosphate. Yeah, so it has to be this actual pyrophosphate.
Oh, okay.
So pyrophosphate can like make the reverse happen and then create another piece of ATP.
Oh, I see. Thank you.
Hello, welcome. There's 1 chair and then there's not chairs. Oh, you're so lucky. Look at you, just for you. Okay, we're coming around this way. Yes, the question.
The NP plus is there in the OA, but it was also there for the stabilization of the
Yes, the incoming DNTP.
Okay.
So stabilize incoming DNTP and then make sure we deprotonate the hydroxyl here. Okay, next, we're going around the circle, I think, okay, skip. Right there.
Why do you hold on?
Okay, cool. Work on my drawing. Okay, so incoming, like you think of your polymerase, it's a right-handed thing, and then your new DNTP is going to come in from the left. And then you have these two magnesiums that coordinate this reaction. Magnesium, magnesium, you have phosphate, phosphate, phosphate. You have O minus here. This is coordinating here, coordinating here, coordinating here. Remember, phosphates have those negative charges, so it's allowing the stabilization of this triphosphate in the active site of the DNA polymerase. And this other magnesium is also helping to deprotonate this oxygen so you can have nucleophilic attack. That look is clear? Okay, cool. So you might be. Okay, keep going this way. Yes, on you.
Oh, okay. This really great. It's something about matching the slash this and you said RNA virus instead of the virus and what's in it. With the activity still being found in the pellet. And I was wondering why it would also be found in the pellet. Because are we detecting like the RNA nucleotides or like are we detecting the protein that the RNA is created? Like is it somehow transferring the label to the protein?
So we use two radio labels in the Hershey Chase experiment. What label is used for the DNA in the original experiment?
Phosphorus.
Phosphorus. Do we have the phosphorus in the same place in RNA as we do in DNA?
Oh, yeah.
Yes. So basically labels it the exact same way. If it was RNA virus, we would have got the same outcome.
I guess what I was confused about though is like if we're doing the sensification, we're talking about like we're talking about the pellet, so I guess it's a nuclear pellet, wouldn't there only be one?
So the pellet is not a nuclear pellet. So we go through the steps. The pellet is the actual bacteria. So in the Hershey Chase experiment, when they have, this is the bacteriophage. Boop, boop, boop. Others. This is our bacteria. And then we label genetic material with pink, which is radio-labeled phosphorus. Usually what happens with a virus is that it injects its content into this bacteria. So the bacteria has DNA, RNA, whatever comes from that virus. We do the whole blender step, blend, blend, blend. We had already given it the genetic material, what was centrifuge. What we end up having is a pellet. A pellet that contains the bacteria that has the genetic material. And a supernatant has the viral particles left behind because they're so much lighter and they don't pellet at the speeds that they use the centrifuge.
Oh, okay, that makes a lot of sense.
Okay, cool. Yes.
I have a question of this too also after. So the blending step is what separates the actual like
Virion or bacteriophage from the bacteria.
from bacteria. So hypothetically, if there was no blending step involved, and we were talking about the one where they labeled the sulfur of the proton dope of like the age, and if there's no blending, does that mean that we would detect the radio-labeled sulfur in the pellet still?
Yes. So if there's no blending, you still have, maybe I'll put it over here.
Is it still be attached?
This will be attached. Right? Still attached. There's a little bacteria. I don't want to put that. Leave the tops off. That makes any sense. And as you have this tube, and if these two are connected, you would see the radio label down here, and then you'd also see the injected radio label. So you wouldn't see any radio label in the supernatant at all, because you did do the blender step, so not everything is at the bottom.
Okay, if like, we only looked at the sulfur, like, we didn't label the phosphorus in that one, you wouldn't even see the
You
pink.
wouldn't even see the pink.
It just looked like if it was this sulfur.
So that blending step is super, super important for interpreting the outcomes or the results. Okay. Yes.
It's still about the same experiment. Instead of doing maybe the
With RNA be possible?
Mhm.
So now it'd be like the lactoviruses and what are.
Exactly, yes.
So would that still be like the exact same thing if the see the RNA?
But yeah, it would be the exact same thing, because the label will label phosphorus and the, yeah, it just labels the gene. Whether it's RNA or DNA, whatever, yes.
Alright, this is unrelated to it, but it's like, what specifically is, if you remind me, what specifically is required for like a plasmid or anything to be like propagated into a cell? Is there anything specifically required to think this to go into the occur or not?
We need for the transformation to occur.
Or I guess, okay, maybe, yeah.
Okay, so for transformation, it's self-type dependent. Bacteria in particular have this ability to take up exogenous DNA just kind of in and of themselves.
Okay.
And many like lab scenarios will like make that efficiency or make a transformation more efficient by say doing like a chemical treatment. We'll do like heat shock that makes like random pores in the bacteria open up. But bacteria is actually a passive thing that will naturally happen. And it's weird because when we first learned about transformation, they knew it happened, but they didn't know how. We still don't completely understand how it happens in bacteria. We just know how to make it more efficient and make it better.
Is there anything you know for the gamma propagation cells with that?
Nope.
Okay, just know that.
Mhm.
Right.
Yeah, no, what transformation is, no, it's separate from like, the replication of the actual plasmid.
Great.
Yes.
I just had a question from the hypothetical you gave of the, like protein was a genetic carrier. Where exactly in the centrifugation would it be found again?
So, If protein was a genetic carrier, you're gonna find whatever genetic carrier would end up inside the bacteria. The genetic carrier is gonna end up in the bacteria.
Oh, and that will always be in the pellet.
Yes.
Oh, okay. Nice. Thank you.
Any more questions about Hershey Chase? Hershey Chase. Okay, yes.
When you say that, like, it's found in the pellet, so would, when DNA like replicates or whatever, is the stuff that's in the pellet, like the DNA itself, or like the proteins that you produce after like it turned into RNA and RNA make like the
It's
protein?
gonna be the DNA itself, but the label only labels the DNA. And the bacteria doesn't have a capability to make like a new label from the protein that was made.
What?
So if we're gonna think about it very deeply, this can't have been like a super, super long experiment. Because over time, the bacteria would take all this, repackage it, make new virions, let it go,
Yeah,
right? So this is like at the beginning of the stage where you just get the input of the DNA or genetic material into the bacteria.
Okay.
Okay, we're gonna start on the outer circle. So we'll go, go ahead, question here.
I'm Cynthia Bowden. You said introduce yourself.
Oh, yeah, nice to meet you.
And I guess my question is from motion food. You mentioned that like without the H1, like part of the histone,
Yeah,
it still functions, right?
yeah,
It's
so
just
functions.
a lot like less compact. And
Exactly.
you mentioned that the chromatin would, or like the nucleus would have to be bigger to contain everything, right?
Yes.
Would that actually impact like the appearance or like the structure of a chromosome or like would it look any different or?
If we had a chromosome, it'd probably be like less ordered, because it's definitely gonna be less compact.
Honestly. So it looked like around, it just. But like there would be a structural like difference.
Yeah, it's gonna definitely impact the structure. So just to repeat, the H1 is necessary for condensation, but you don't technically impact the function. If you think about downstream and like in more detail, you can impact the ability of other transcription factors and things to access the DNA. So it could make more genes accessible. It may change the genotype of the cell, but in and of itself, just not having H1 doesn't like kill a cell or anything like that. You can still use the DNA, you can still replicate the DNA, you can still do regulation by how you control what's on the histone tails.
So you still look the same, but not exactly.
But not exactly the same, yeah,
Thank you.
yeah. So know its role in compaction of the DNA. Yes.
So, there's this thing about histones and stuff, so when, histones interact with DNA, does it use the quality of it, or does the charge?
Charge number one, charge interaction is the most important thing for the DNA to interact with the histones. The fold interaction, basically that spacing of about 10 base pairs, having like 2 A to T's helps it bend and fold tighter, and you get better, more compact association with the histone depending on that pattern. But no matter the pattern, you are going to get association of histones with the DNA because of the negative charge.
Yes, so that's right, so this is the connection between the DNA and the histone, the nucleosome are, is mainly between the conserved histone fold and the phosphate backbone, not between charges specifically.
That's the positioning. So the question is asking what parts are associated.
Oh, okay. All right.
Yes.
I thought it was histone one, should I expect to see more synthesis occurring since it's going to be after that?
So that's possible, but you don't have enough information to actually tell. Yes,
It's like, it's like, for like, it's like wrap around, it's doing these like very, like being like more 18 weeks.
yeah, so the repeats is basically, say it's about 10 base pairs. Gosh, 10 base pairs, and usually have about 2 A to T's. Every 10. These motifs actually make for the most tight coils like around the histone core.
It's like maybe in a place where like,
Accessible at all, like they,
less.
yeah. Yeah, I mean, usually you don't see DNA accessibility regulated by this coiling. DNA accessibility really much is regulated by post-translational modifications. Usually on the tails, yeah. Okay, yes,
I mean, it's definitely no one, the types that are about post-translational, like, not to be seen.
nope,
Oh, and another thing, why is it like?
so after I got access in class, I went to go look up the reason that a student brought up, and the reason was actually correct. So because of the steric hindrance of a G to C pair that has 3 bonds, it's harder for it to bend. There's less steric hindrance for the 2 hydrogen bonds, so it's easier to cause a bend when there's like more, it's almost like a hinge, right? And so if you have a big bulky thing, it's harder to make it the bend, and it's smaller, it bends a little easier. Okay, so I think we're falling to the outer rim. Is that a movie? Not specific rim. Okay, let's go here.
There's just a question in the videos, because I just like all this true. This is where it's possible to have it
RNA to RNA should be true. Was that one that was mismarked? Okay, which one?
I think he's like laughing and it's supposed to be.
Yeah.
But I don't know if you know one.
Yeah, let me double check it. What question number is it? I just want to make sure it's marked right for everybody so nobody gets confused. Yeah, so RNA to RNA hybridization is 100% true and that can happen. So you said it's question 11 on, okay. Yeah, got that. Okay,
What's the place of this one on?
it's 2. Yeah, so FYI, so everyone knows, whenever there's like a mismarked question or whatever, everybody will get the points back. Like y'all not gonna get dinged for my mistakes, that's not fair. Okay, let's keep going around the circle. Yes.
I was just thinking on how. You said that they will be identical, but I thought they were. Because of the economy. My logic is that large terms from like, but it's not getting the proper copy of it. Right? It's not like
Yeah.
it's a proper copy, so they're dying off, so that's why they're smaller economy. I think you mentioned that they would all be identical in some sense.
Okay, so I guess what we can do is talk about like how colonies are talked about in lab versus technicalities, and I don't want us to get too hung up on this. And so what we're saying is that if you have you don't have a centromere and you basically don't have equal division and you have one plasmid that ends up a cell that gets 2 of these. So say it's a cell, I had 1 plasmid, I tried to divide, and then I ended up keeping my 2 plasmids, and then I try to divide again. And then this time it divides correctly and I get cells of 1 plasmid that they're not technically genetically identical, but they have the same set of genes and they're all going to produce, they just have more copy number. And so we would still consider them being genetically identical because they're producing the same genes, even though they'll have different copy numbers of that plasmid. But don't get like hung up on that. When we think of colony is the same set of genes that are going to get made and expressed. Yes.
That's what the on is, so, is it the, what occurs, sorry, what ensures that replication occurs at a particular number of times is essential, correct?
Say the question again.
What ensures that replication occurs at an appropriate number of times?
Well, centromere ensures that you have like good separation of the chromosomes. So equal separation every time. So every daughter cell gets 1 copy.
Yeah.
That's what the centromere is for.
So then what ensures, the tissue replication, because probably number of times just the percent efficiency.
So if you think of every organism has their own like level of replication, so the way the question is worded is kind of like organism specific. So we don't really ensure that by like the type of plasma that we give it. It's gonna replicate the way that it replicates based off of like the environment it has around it, the nutrients that it has. Because that's how we control it, how something will grow or how fast it will grow, how slow, whatever. It's what nutrients that we make available to it. Like what the pH is, so like all the conditions. And so really what we're thinking about with the centromere is just making sure that every daughter cell gets the right number of chromosomes or the right number of plasmid. Yes.
Each colony is like, colony time is 100 cells, like replicate cell, or this is other cell, so then like it's sort of like 15, like each one of them, they have like 15 separate
Colonies, yes, yes.
Reading the, like, the two number, there's the one that, like, how many bands did you see to extract the DNA was in acidic conditions? I put, like, greater, but I just want to make sure that my thought process behind it is like.
Okay. So what I'm gonna do, I'm gonna draw this. Because I actually hate this representation of gels because it's a little bit confusing. I don't use this representation on the exam. But it gets the point across. But this is actually a lot more representative of what you'd see in a real experiment, as opposed to like the pretty arts.
Yeah.
That that we tend to use for the exam. Y'all remember this question? You see what I'm drawing? Okay. Walk me through your logic.
Okay, so this was asking, about the number of bands that we would actually see
Mhm.
after a round of replication. And so I thought that it would be greater because in acidic conditions, the DNA would like start to. Would it start to come apart a little bit?
What does it say for the acidity?
Acidity, very acidic conditions.
Okay,
So I assume that because it's like not going to be all like nice and put together like it normally is, that it would result in like.
and more bands start
more
breaking down.
bands.
Yeah,
It would start separating.
okay. Spent a lot of time drawing.
Yeah, sorry, I guess this question's more relevant to your drawing, to your drew it out, but, so. Okay, in this, they have like. But, it's like the lanes that they're in, right? It's
Mhm.
asking for which one would be after one round replication. And I know it's 4 because like, it's like, oh, it's all in like the same area, and it's all mixed. But what if the mix would be like? Or does the actual positioning of it matter?
The
Like,
position
left to
matters.
right?
So what does this figure say about what is left and what is right?
It says,
What does it say times 0 is?
N 14 is at T0.
Okay, N14 is at T0. Does it tell you anything about like which lane is T0 or T0 is left or right?
That was the question before that, but, it was that, T0 was left.
Okay, so if T0 is left, that means whatever we're starting off with the parent strand is the furthest left.
Yes.
Okay.
So
Mm,
would it like,
So one round of replication. The left should be the heaviest, right? And then so if you have one round of replication, then you're gonna incorporate whatever the opposite is,
so
and then
will you shift?
so it's shifting.
you know,
And then based off of whether it's going to be a what you call it, a native gel or an alkaline gel, we'll know if we're supposed to see one band or two bands. So what does the question say about the condition for the gel?
It didn't, the condition, oh, it's a native,
Okay, so it's native, so it's all together. We had one round, so you're gonna see one band.
one there.
Mhm. So you're looking for one band that's shifted from wherever time
Wherever
0
the
is to something else.
Oh, okay.
Okay, does that make sense? How you think about that question?
Because in the homework, there's like 2 images of one band, right? But one of them's on answer choice.
Yes,
So I was just like, oh, it has to be this band, but I didn't understand.
but the why.
Yeah.
So think of time 0, figure out where time 0 is. So another way to think of, remember I said, or whatever you wrote in first, that's gonna be parental strand. whatever's times 0 is also parental strand. Right, and then once you have parental strand, you know, anything that shifts or moves is whatever the label is for the daughter strands. Then native, after 1 round, it's all gonna be together. Remember I said, take your time to draw them. And then so for that, the answer would end up being like it all shifted together. It's lighter. It's number 1. Okay, questions? Okay, let's keep going. Where are we? Yes, we're over here somewhere. Question.
Oh, yeah.
Okay, yes.
For RNA primers, am I only gonna use it to synthesize like these fragments, or do I use it on the?
For Okazaki fragments. And so, like I said, we're coming back to it. It's not going to be on exam 1, it would be on exam 2.
Okay.
Yes,
CGL primers for the particular sphere that you just synthesize it immediately.
I'm 99% sure. Don't
Okay.
quote me.
Yeah, because the both kind of diagram.
I thought I had a diagram where it goes back and like eats it up,
Yeah.
like eats it and comes back behind it. So I think you have it on, yeah,
Yeah, I understand.
on the illustration. So we'll double check it, we'll make sure it's accurate. Just notice on exam 1, we're coming back to it. I think right after exam 1. Oh, it's coming, y'all. It's coming. Okay, yes.
As a quick question, the video that was perfect with for number 4 was regarding whether ways that we just in general about nucleases and proofreading and pyrophosphorus. Just in general, so proofreading occurs with is removed by a 3 times 5 times X nuclease, correct?
Yes.
And then you said a question is DNA polymerase can discriminate against a wrong base pair but make errors. What type of exonuclease activity fixes these errors? Is the 3 times 5 times X.
Yes.
And for endonucleases versus exonucleases, you just want to say like the outer versus inner, just know that. And then
100%.
between 3 times and 5 times and 5 times and 3 times, just know like the direction.
Direction,
Differences.
yes.
Okay, and then Nick translation, that's not going to be on this.
No,
exam. I mean, I read, I'm aware of it, but just.
Yes, but not on this exam.
Thank you.
Okay, cool. Good here. Yes, so if you think about a probe, probes are single stranded. And then so I want you to remember that RNA is actually very susceptible to hydrolysis, either in acidic conditions or in high temperature. I think this is not directly clear in your reading, so I'm going to say and reiterate it, that high temperature is not good for RNA probes. As acid is not good for RNA probes. And then so when you're in a condition that is not optimal, you choose a DNA probe over an RNA probe. Otherwise, the RNA is stronger and has better like affinity interaction with DNA. Okay. Okay, this side. Any questions on the end here? Mhm. Yes. Yeah, so the reason why it's no, I mean, it's not no, it's because it's not dependent on a specific sequence. Any DNA will wrap around the histone core, whether or not it has that pattern. And then the reason why we don't say yes is for the same reason. You don't have a specific sequence of DNA that allows it to wrap around the histone core. All DNA is going to wrap around it, but it does get optimized if you have that pattern of 10 base pairs and then A to T's every 10 base pairs. That's why we say sort of. Okay, I'm not going to ask you a question on the exam that's a yes, no, or sort of. That's kind of weird. But of course, your lecturer wants you to think through it. Okay, on this side. More? Okay, free for all.
Well, I had a quick one regarding on the exam. I say like select all high question. We never kind of select all that high question, but only one of them is correct. And typically would.
Sometimes I would, but it's rare. Usually select all that apply will have multiple. And then like I don't want to stress y'all out, but with the select all apply, you'll normally see that they're actually worth more points than other questions. So there's multiple answers. Like I can't hide the point value. I tried that, Cams doesn't let it happen. So you'll see the point values for every single question. So don't get lost in trying to deduce how many answers based off of the point score. Sometimes like it's 2 points to answer, or 1 point to answer, or 3 points to answer. Like you don't know. So don't spend your time trying to do that. Instead, read through each one, ask yourself, is this true or not? Am I 100% confident in the answer? If you are, then mark it. If you have any doubts, leave it unmarked. I'd rather you get an answer wrong and not get the points than the mark an answer wrong and get subtracted for stuff you didn't know. So only mark what you are sure of on those types of questions. Clear? Okay. Those types of questions can suck, but you can only get them, yeah, but you get them, if you understand the concept, you will get them right. Okay, any other major questions? Yes.
Going back on the codes. So you would want to do RNA if you do that optimal,
Mhm,
and DNA with it.
correct.
Why are you become so more typically used to be?
Yeah, it's a stronger approach. So basically, I wouldn't say there's a typical, every lab kind of chooses what they like to do. So even though like probes are stronger interactions for hybridization, some labs will be like, let's just do DNA because it's stable, we can store it forever, we're not worried about it getting messed up. So people make decisions based off of their own conditions, but just know that an RNA probe is stronger. So the way I'll ask the question is, which probe will hybridize the most strongly? Which one is the best choice for this experiment? And that's how I word the questions. So RNA, the strongest, the best, unless there's not good conditions that will cause it to get hydrolyzed, then pick DNA. Yes.
Could you elaborate on the 5' and 3' and because I was looking at the diagram and it looks like it might be located in one of this domain. I was just a little confused why it would be like in the places it's going to have about the actual like domains itself.
So it's not actually like working ahead. Let's see if I can draw this out. Let's call this A, T, G, C, A, and then the new nucleotides come in from the left. I'm sorry, let's do. Okay, we're just gonna draw it in. This is the active site. And then so basically you're adding in a new and TP, and then you want to connect it here to the 3' end, because we always synthesize 5' to 3' in that direction. And then so what happens is that you have exonuclease sites that aren't like right here at the top by the finger. That's actually where you have the extension, but you have one that's down here, if I'm correct. There's a 3' to 5' exonuclease. And then when it detects that a nuclease is wrong, it literally kind of like lifts off and repositions itself so that now you can go with this active site, back it up, erase it, and then it flips back and then it goes forward again. So it literally changes the active site. And then so what's interesting about the way this works, because we have two different there's a video I'm going to show you after exam 1 where these polymerases actually act in complexes. So to make the whole machinery be more efficient, you don't necessarily have to have the polymerase that has the active site on its own actually do the activity. It can move out the way and let one that's kind of over here come and do the activity, and then the other one keeps going.
Okay.
So that video kind of helped clarify. Usually you have like a couple of 3 polymerases kind of helping each other out and doing a longer replication for.
Okay.
Okay. Any other questions?
What's up with the leading strand requires a primer, it says that it requires like one primer and then.
Upton has multiple.
Yeah.
Yes, she is right. Yes. Any other questions?
How much of the analogy do you want to think about?
None.
None. And then, with the talks about DNA polymerase added into that to the 3' end of DNA, we kind of just went through that idea of with the magnesium.
Yes.
Okay.
Yes, question.
Is there a limit on how many times an exonuclease can work? So I know it was talking about it sees the error, it feels like a correct 392
Mhm.
I feel like does it overview it like the first time, or is it kind of just like grab it out like?
I don't know. Yeah, I mean, I don't know, because it's basically, it's the stoichiometry and then association and affinity. And then usually the right base pair has better affinity, so it'll match up. But I mean, I could imagine it could make a wrong mistake twice in a row. Like, I don't see why. It's not impossible. Yeah, sorry if that's not a satisfying answer.
No, that's more than.
Okay, yeah.
Go back to what you mentioned, like, this was that, orders of replication, telomeres and centromeres are unique characteristics for mainly eukaryotic cells in DNA, not bacteria.
Yes. So mainly
Bacteria is
characteristics.
similar to order of replication, but not quite.
Yeah, but not quite. Yeah. And so the most important difference is actually gonna be the telomere, and then once again, we're coming back to telomere after the exam, so I keep like dropping in telomeres. And then we'll come, like, spend a lot of time with it, in some of the subsequent lectures. That's the main difference where like you have a eukaryotic and not prokaryotic. And then you have these similarities and concepts based off of like centromere and the origin of replication. Bacteria said have like a start site, transcription start site. And then other cores are sets of proteins that allow the genetic material to evenly separate between the daughter cells. Yes,
This polymer is actually even distributed, this is the top right. We just focus on processes.
there's this process of synthesis. And we had a hand here.
A lot like the series, there's another point on it, but if it were to be like heavily incorporated into a plasmid, they wouldn't have any effect.
It wouldn't have any effect.
Okay,
So
it wouldn't
if
be-
a plasmid is circular,
Mhm.
putting telomeres in is not gonna do nothing. It's just gonna be sequences and repeated in there. If you have a linear plasmid, which is almost never used, you have to have telomeres in order for it to work.
So it wouldn't like affect the original function of the plasmid by itself.
No. We use the sequences. Yes,
I was wondering, it kind of going back to what I mentioned before, question, but it was a specific question on the one of the frequencies. It was saying how like the I covered it today, but I just want to go over it again, was like the contact between the DNA and some of the nucleosome are, and they give it a list, but the one that I was confused about was says is it mainly between the R groups, the histones, and the phosphate backbone of the DNA, which is incorrect. And then, but I guess the correct one is that it's conservative in how it folds, but how come it's not the R groups and the histones with the backbone? How come that's not a way of the contact?
the R groups aren't where you have that positive charge from the proteins, from the amino acids. If I'm correct,
Oh, it's the R groups of the histones.
of the histones. Yeah, so it should be because of the charge. It says R,
This is R groups of histones and the phosphate backbone of DNA. That's the
reaction.
it was incorrect. What it is it?
I thought it was technically kind of good, correct. I'm gonna look at that one. And I also send like, so anything that sounds kind of weird like that, I'll put it as a post and make sure everyone sees it. But I'll double check that so it doesn't confuse anybody.
But just so that we know, between histones and DNA, the main fraction is charges as well as folding with AC and days.
Yes, those two main
But what's the
concepts.
strongest you would say is?
It's charge. Charge is what keeps it associated.
Thank you.
Okay, but I'll still go check that question. Yeah, it can be hard to remember what I wrote in my head, like a month ago, so I'll go read it. Yes,
Is we be able to differentiate between two middle island and something that's in
nope,
it's not one myself.
yeah, no diplomers. Okay, any other questions? Yes,
It was a question about the Plasmodium minor, where it was saying, when we want to, basically the question was that, the difference between the mutant and the wild type genes for this, from a person with Plasmodium minor can be covered by examining the HPV what.
Mhm.
And looking at with mRNA and DNA, which I can see why I can do actually structural differences between them, and obviously mRNA is a clone of the DNA, so you can still see the differences. But the reason why protein is incorrect, I just want to be sure, is because that's like beta Plasmodium minor people as well as normal or wild type people will still show protein.
it's also protein, it's the same protein, you just have less. So the actual mutation that we wrote in is one that basically, I said that it's, what do you call it when half of your chromosome? Heterozygous.
Heterozygous.
Yes, it's heterozygous, so mutation is only like in half, so you have a chromosome that expresses all the normal everything. So the only way to identify is by looking for changes in sequence. Because the one that has the mutation is not making
So if you
protein.
said amount of protein, then that would be.
Yes, I said amount of protein less, and I actually got that thing in there.
Okay.
Okay, any other questions? Okay, so I'm gonna reiterate. Go over your notes first, everything that you've gone through, make sure you feel clear on all the learning objectives, and then sit down with space and time where you have a block of like an hour at least, then take the practice exam. Take it as if it's a real exam. And then see what you got wrong and map out the concepts that are like, okay, I wasn't clear on that concept, then use those to study. Yes,
One time for practices now.
no, unlimited. You can see all the answers.
We don't get graded.
No grades, no points, no nothing. So that's why because after you go through it once, you already start building like muscle memory of the questions. So make sure that time that you go through it, you maximize like knowing and learning what you don't know. And then, we're in here for Monday at 8 AM. If it's too full, at 9 o'clock, we'll move over to where discussion section is. Okay, I will post this and then I will see y'all on Monday.
Thanks, Jose.
All right, bye.